A microscopic cut factory turns dietary fiber into short chain fatty acids that travel via bloodstream to modify histone acetylation and regulate gene expression. The colon hosts a diverse community of microbes that digest complex fibers. These bacteria break down polysaccharides into simpler sugars through enzymatic pathways. Fermentation of these sugars produces short chain fatty acids, mainly acetate, propionate, and butyrate. Acetate is released first and can be absorbed by the host for energy production. Propionate follows, traveling to the liver where it influences gluconeogenesis. Butyrate accumulates in the colon lumen and serves as a primary fuel source for epithelial cells. Bacteroidetes are efficient at degrading plant fibers into acetate and propionate. Firmicutes specialize in producing butyrate from resistant starches and other substrates. The balance of these species determines the relative concentrations of each SCFA. Higher butyrate levels correlate with improved colonic barrier integrity and inflammatory effects. Acetate and propionate modulate systemic metabolism through signaling pathways in distant tissues. Thus, microbial fermentation shapes host physiology by delivering specific fatty acids to target cells. Pause 0.8. Bacteria first convert glucose to pyruvate through the glycolytic pathway. Pyruvate then enters the acetyl-CoA branch via pyruvate oxidase. Acetyl-CoA is released by phosphotransacetylase and acetate kinase in the cytoplasm. This produces acetate, the simplest short-chain fatty acid, and released into the lumen. For propionate, bacteria use a different route, starting with lactate. Lactate is oxidized to pyruvate and then carboxylated to methylmalonyl-CoA by a carboxylase enzyme. Methylmalonyl-CoA is rearranged into propionyl-CoA by a racemase. Propionyl-CoA then becomes propionate via a decarboxylating enzyme. Butyrate synthesis begins with two acetyl-CoA molecules condensing. The condensation product is acetoacetyl-CoA, then reduced to butyryl-CoA. Finally, butyryl-CoA is converted to butyrate by a thioesterase. These SCFA are then released into the gut lumen for host interaction. Pause 0.8. Short-chain fatty acids are produced by gut bacteria in the colon. They must cross the epithelial barrier before entering circulation. The primary transporters are monocarboxylate transporter 1, MCT1, sodium-dependent monocarboxylate transporter 1, SMCT1. Also facilitates uptake. Both transporters use proton or sodium gradients to move SCFA into cells. Once inside enterocytes, SCFA are released into the portal vein. The liver receives the first pass blood and can metabolize a portion of SCFA. Surplus SCFA escape hepatic metabolism and enter systemic circulation. They then distribute to peripheral tissues, including muscle and adipose tissue. In these cells, SCFA act as signaling molecules that influence gene expression. Specifically, they modulate histone acetylation by inhibiting deacetylases. Pause 0.8. Short chain fatty acids are produced by gut bacteria during fermentation. The most common SCFA are acetate, propionate, and butyrate. These molecules diffuse into nearby colonocytes through passive transport. Once inside the cell, they can bind to histone deacetylases. Binding of an SCFA blocks the catalytic pocket of HDAC. Inhibition of HDAC activity reduces removal of acetyl groups from lysine residues. Consequently, histones remain in a more acetylated state. Acetylation loosens chromatin structure and promotes transcription factor access. SCFA also enhance the activity of histone acetyltransferases. This dual action amplifies the signal for gene expression changes. The net effect is a shift toward an open chromatin state. These epigenetic changes influence immune cell function and gut homeostasis. Pause 0.8. Short-chain fatty acids produced by gut bacteria enter colon cells. They inhibit histone deacetylases, increasing acetyl groups on lysine residues. Acetylation neutralizes positive charges on histones, loosening DNA winding. Looser chromatin allows transcription factors to bind promoter regions more readily. RNA polymerase Roman II is then recruited to the transcription start site. This assembly initiates RNA synthesis for target genes such as IL-10. IL-10 mRNA levels rise, promoting anti-inflammatory signaling in nearby cells. Similarly, PPAR gamma expression increases, influencing lipid metabolism pathways. The overall effect is a shift toward a more regulated immune environment. These changes illustrate how metabolic signals from the microbiome can directly alter gene activity. Thus, histone acetylation acts as a bridge between bacterial metabolites and host transcriptional programs. Understanding this link helps clarify mechanisms of gut-brain communication and systemic health. Pause 0.8. Gut bacteria ferment dietary fibers into short-chain fatty acids, mainly acetate, propionate, and butyrate. These metabolites diffuse across the intestinal epithelium and enter systemic circulation. In immune cells, they act as ligands for G protein-coupled receptors. Binding activates downstream signaling that increases histone acetyltransferase activity. Enhanced acetylation loosens chromatin structure and promotes transcription of anti-inflammatory genes. One key outcome is the differentiation of Na-A-tilde-V T cells into regulatory T cells in gut mucosa. Regulatory T cells secrete cytokines that dampen excessive immune responses and maintain tolerance. In adipose tissue, SCFAs stimulate acetylation of transcription factors controlling lipogenesis. This shifts lipid storage toward healthier fatty acid profiles and improves insulin sensitivity. Collectively, these epigenetic changes link diet, microbiota, and host metabolism. Pause 0.8. Recent studies show that gut bacteria produce short-urose chain fatty acids SCFAs, which can enter host cells. Once inside, SCFA act as substrates for histone acetyltransferases, increasing acetylation marks on chromatin. This epigenetic change loosens DNA winding, allowing transcription factors to access specific genes. However, the sensitivity of different HDAC isoforms to SCFA derived acetyl groups varies across tissues. In liver cells, class I HDAC appear more responsive than in urinal tissue, but data are limited. Long I term exposure studies are scarce. We do not yet know if chronic SCFA levels sustain or reverse histone acetylation patterns. Another gap lies in the spatial distribution of SCFA within the gut lumen and how this influences local versus systemic epigenetic effects. Future experiments should map SCFA gradients using mass spectrometry imaging, while simultaneously profiling HDAC activity. Additionally, singly a cell attack a sec could reveal cella type-specific chromatin accessibility changes after SCFA treatment. These approaches will clarify whether SCFA preferentially targets certain gene networks in distinct tissues. Understanding these mechanisms is essential for designing microbiome-based interventions that modulate epigenetics safely. Pause 0.8. Gut bacteria convert fiber into acetate, propionate, and butyrate. These SCFA are absorbed, circulate, inhibit HDAC, increase histone acetylation, open chromatin for transcription of genes such as ILA-10, thereby influencing TREG differentiation and metabolic pathways.